Good afternoon. My name is Zachary Kennedy, and I'm with my friend. What's your name, boy?
Matt Russo.
And this is my human organism final exam project. I'm here to interview Matt. He works at a biotech company based out of Cambridge called Acceleron Pharmacy. So Matt, what is your job title at Acceleron?
Well, I am a research assistant in the antibody technology group, which falls in the research and development division of our company.
Oh, very, very cool. And we're here to talk specifically about a drug protein you have, which is in clinical trials,
Yes.
called ACE31.
ACE31.
Yeah. Tell me a little bit about that.
Well, ACE31 is a drug. It just entered phase 2 clinical trials.
And what does that mean?
Well, that means there's 3 phases in clinical trials after it goes from preclinical, which is mice. So phase 1 are kind of broad patient studies. Phase 2 is a little more intensive with you get larger numbers of people involved, and you start to really specify your variables in your treatments and your treatment plan. And then phase 3 is just, if phase 2 and phase 1 look really good, you do further testing in patients, and they're all generally the same principle. You're trying to make sure your drug is safe, your drug is effective, and that's pretty much it. And then after it passes phase 3, then it goes for a final evaluation by the FDA before eventually going on market.
Okay, very, very cool. And so this drug, what is it cure, or what does it treat?
Okay, so ACE31 is a muscle-producing drug, and it will be used for patients with ALS and muscular dystrophies.
Wow, that's very cool. And on what level in the human body does it work? Is this a protein drug? Does this work to rearrange the DNA?
Well, it does a little bit of both. So what it actually is a fusion protein.
Okay.
You know what a fusion protein is?
No, what is a fusion protein?
Well, fusion protein is, you're familiar with the general structure of a transmembrane receptor protein?
Okay. Yes, a receptor protein is something that sticks out of the cell and is able to be recognized
Yes.
by other objects in the body.
Yes, it does.
Okay.
So you have the 3 parts: the extracellular domain of the receptor where the ligand actually binds to induce a response. Then you have the transmembrane section, which is embedded in the membrane. And then you have the intercellular membrane, which is usually a little piece right inside the membrane, which induces the pathway. So fusion proteins take the extracellular domain where the ligand binds, and it takes only that section, and it fuses it with the Fc portion of a human antibody. So what you get is a soluble, what we call decoy receptor, that it's a soluble protein that is free floating, and what it does is it attacks the ligands for that receptor that you took the extracellular domain off of. So instead of binding to the normal receptor that this ligand binds to, which is embedded in the cell and induces the regular response, it will bind to the decoy receptor, therefore inhibiting whatever pathway that the ligand induced when it bound to the normal receptor.
Okay, cool. And specifically, what do these decoy receptors, what do they target?
Well, you mean specifically ACE31?
Yes, specifically ACE31.
Okay, so ACE31, it is fused, the Fc portion of a human antibody is fused with the extracellular domain of the activin R2b receptor. And what that is, it's a receptor that myostatin binds to, and when myostatin binds to this receptor, it inhibits muscle growth. So the whole plan with ACE31 is that you take the portion of the receptor that binds tightly to myostatin and you fuse it with a human antibody, FC, and so what you get is you get the decoy protein that swims around, floats around probably, and whatever myostatin is there will bind to the ACE31 instead of the activin R2b receptor, and therefore the pathway and cascade signaling that will be induced that will eventually lead down to, you know, inhibiting the genetic expression of genes that control muscle growth, aka inhibiting them. That whole pathway will be inhibited. So, in essence, you're inhibiting the inhibition of muscle growth, therefore promoting muscle growth.
So to sum up, your drug gets injected into the body, right? It steals or takes up all the myostatin, and when the myostatin is not received by the other receptors on the cell, inhibition doesn't occur and growth continues uninhibited.
Exactly.
Oh, very, very, very
That's
cool.
the goal.
Now, it's very interesting. However, I can foresee some problems in society. Do you foresee perhaps people using this drug, abusing this drug, genetically engineering themselves to be bigger, stronger, faster?